Buenos días a todas y a todos. Bienvenidos a esta maravillosa ciudad que es Elche. Estamos muy contentos y muy orgullosos de haber podido reunir en este espacio a gente tan notable como los investigadores que van a estar en este programa y los invitados que tenemos, todos vosotros. Estamos encantados de recibiros aquí. Y yo quiero agradecer a todas las personas que han hecho posible este congreso. En primer lugar, al personal de la Fundación 100, especialmente al personal de gestión, que se han volcado, que han estado trabajando incansables hasta los domingos y, bueno, que se han implicado muchísimo en esto. Por supuesto, a los sponsors, a la Sociedad Española de Neurología, y al Ayuntamiento de Elche, que también ha echado el resto y se han portado genial. Nos han cedido este espacio que es maravilloso y el Palacio de Congresos, y ha tenido una implicación absoluta tanto el alcalde como el resto de la corporación local. Y bueno, pues muchísimas gracias a todos. Esperamos que disfrutéis mucho con este congreso y voy a dar paso a Javier, que ha sido otra de las personas que ha hecho posible este congreso, que ha estado apoyando desde el minuto 1 y que se ha implicado muchísimo. Javier.
Buenos días a todos. Yo creo que como casi todos nos conocemos y conocemos a esta gente, sabemos que no es ninguna sorpresa que las cosas vayan a salir bien. La implicación de la Fundación 100, de Pasqual, de María Ángeles, ha sido como siempre encomiable, y el apoyo del Ayuntamiento desde luego a destacar. Yo, para los que no lo sepáis, además de que trabajo en la Universidad Miguel Hernández, soy ilicitano. Así que para mí es verdaderamente una alegría poder acogeros en mi ciudad. Espero que todos os llevéis un recuerdo maravilloso y a vuestra disposición para cualquier cosa que haga falta. Es una oportunidad encontrarnos de nuevo, y con el deseo de que tengamos tiempo para charlar todos, estoy seguro de que, aparte del espléndido contenido científico de la conferencia, la oportunidad de interactuar pues va a hacer que nos vayamos todavía con un mayor recuerdo. Muchas gracias. Pasqual.
Bueno, yo soy licitano también. Benvinguts a Elche. Bienvenidos a Elche. It's a really great pleasure to have you here, to have such a wonderful speakers coming from all over the world to my hometown. I'm really, really excited, and really excited also because we are going to talk about treatments, and this is something that 10 years ago, 20 years ago would be something crazy if you think, oh, I'm going to dedicate my congress or one Congress of the year to treatment, but I think this is a moment of hope, and this is what we want to really to show here, that there is not only very interesting preclinical advances, but also clinical advances. And this is what we have tried to do here, mixing neurologists with basic researchers, or even neurologists that will talk about mechanism and show what is going on in the field, which I think is hopeful, and well, try to enjoy and learn of these days. And for that, we have one of the best speakers in the world. And to start, nothing better than with Mercè, our dear Mercè, who will be chairing the first table. Thank you very much.
Good morning, everyone. Good morning. Thank you very much for to be here. And moltíssimes gràcies a la ciutat d'Elche i a tots els que enteneu aquest llenguatge tan maco que ha estat parlant en Pasqual. Mi agradecimiento a Fundación 100 y a la Fundación Reina Sofía por haberme invitado a esta ponencia, que yo pienso que será muy interesante, interesantísima, very interesting, so interesting, and my personal agreement and a gratitude to the Pasqual in order to invite me to be here and present this first session. Vamos a hablar de Tau. Tau. What is Tau? For me, Tau is the Cinderella. Always was the Cenicienta. Nobody was interested in Tau for a long, long time. 75, discovered, 80s, to link to the Alzheimer's disease, and a little, and a little, and a little. And now, beta is a diva, but tau maybe is the best colors in the world. No? And in this panel, we talk about the colors. And all the project from here is how its tau became as important protein as a biomarker in order to predict and maybe to follow and monitoring the clinical trials of the patients. I don't know. That is your role. No, that is your role, Marc. Marc is a researcher from the Fundación Pasqual, Maragall, and Beta Research. And he's a good and a fantastic curriculum here in Barcelona, but around the world. And he was a good training as a neurochemistry with the group of the Göteborg, and is one of the leaders of biomarkers in Alzheimer's disease. The other thing was, what is tau in the middle of the synapses? And my good friend, that Alberto, that is the chief of the Department of Neurology of the Hospital de Sant Pau and the chief of the Memory Clinic in the Hospital de Sant Pau, I remember that your PhD was about the synuclein? No?
Genetics.
Okay, and genetics. And he leads this fantastic program and promote that Hospital Sant Pau was one of the leaders in Spain and in the world in the neurodegenerative disease, and he wants to talk about synapses. No? What is the role of the tau in the synapse? Is another role, biomarkers, synapses, and prions. No? That is fantastic. You know that, and thank you so much to be here and to present, I believe, something so interesting, because tau have a new role, or a possible new role, in order to understand how its development in the time, the process of the Alzheimer's disease. It's completely different from the beta. Beta accumulate, and the other, it's moving. And thank you so much to be here and to teach about this kind of the novelties. And finalmente, nuestra amiga que está dedicada a el transporte y la electrofisiología de las sinapsis, y va a hablarnos de how tau hits in the middle of the resilience in Alzheimer's disease. Thank you so much. That is their time. In the domestic rules is at the end of every one of the speech, back here, and we are in the TV. Okay? Please, Marc, all is for you.
Thank you, Mercè. Have one here. Okay. Good morning, everyone. Thank you, Mercè, for the presentation, and thank you, Fundación 100, for inviting me here. It's really a pleasure. So I'm starting talking about tau, and I will focus on my field, which is biomarkers. I think that at this time, all of us, we know a lot about phospho-tau biomarkers, especially phospho-tau 217, that actually many of us, we are already using it in our clinics. But I would like to do also a kind of a historical perspective, because this is something that now we assume that we are using, all of us, but it's been really in a short time that things have changed a lot in the field of biomarkers. Actually, it was some years ago quite skeptical that at some time we would have a plasma biomarker able to diagnose Alzheimer's, and now we have it. And this is a good example of things how can change in the biomarkers field and in the Alzheimer's fields, and how this can be translated into the patients. So these are my disclosures, and let's start from the very beginning. So why we have now blood biomarkers? Why now? Because for a long time we didn't have the opportunity to have these biomarkers, and we were all relying on CSF biomarkers, which is still the standard of tooth, and amyloid PET. I would say there are 2 or 3 things that have done the change, and it's a good example for other areas of research of Alzheimer's disease. First of all is the technology. So there are new technologies, and as soon as there is a new technology, I think that we have to take the opportunity for that. Because one of the things is the ultra-sensitivity assays. Look at this. This is, from ALS Forum. And here there is a meta-analysis of the different assays of plasma phospho-tau 181. Most of them were quite bad. But there was a change here let me show it here. It's important to remember that the first papers. The first paper was from Michel Milken in 2018. And look at this. It was the first time that phospho-tau 181 showed that was in plasma a little bit increase in Alzheimer's disease compared to controls. It was not a big effect size. That was an MSD-based assay. And in that case, we could see that there was something. And 2 years later, Tommy Karikari in Gothenburg showed in a Simoa-based assay that we can measure phospho-tau 181 in a much better way. And here we already see that there was an increase in plasma phospho-tau more clearly. At that time, we also did the same in the ALPHA study, which is a study in preclinical Alzheimer's disease in Barcelonaβeta, and we could show that phospho-tau 181, although not with a big effect size, also increases in preclinical Alzheimer's diseases. So, as I was saying before, one of the big changes was the ultrasensitive platform. So now we have technologies, and now actually we are increasing, having more technologies. We have all the proteomics platforms, SomaScan, Olink, Alamar, and we have to take opportunity of these platforms. But at that time, we had platforms like Simoa, MSD, now Alexis, and also, of course, the mass spec. And that allowed us to measure things that were difficult before to measure. I put this example taken from Kai Bleno. So the concentration of phospho-tau in blood is much lower than the one that we find in CSF. It's 100 times. Just to have an idea, we can compare the concentration with a sugar cube diluted in a pool, in an Olympic pool, and for the CSF concentration of tau, we would have to take 1/10 of this sugar cube and dilute it in the swimming pool. That is the concentration of tau in CSF. But in blood, it's much, much, much lower. We should have to take 1,000 of this sugar cube and dilute it in the swimming pool. And this is the concentration of tau in blood. So it's extremely, extremely low. So that's why we need ultra-sensitive technologies to measure tau in blood that we were not able before to do so. Or sometimes we found some signal, but it was not robust enough. The other thing is the biochemistry. And actually, one of the things that I want to, in this talk, highlight is the importance of the classical biochemistry, because there are some basic things that are extremely key in research. Most of the assays that were, this is the protein tau here, and most of the assays, you can see it in the upper part, that were used in the CSF assays, were using antibodies against the Mead region of tau. But later on, we know with biochemistry studies, especially with mass spec, that most of the fragments of tau that we found in blood were N-terminal, so this part of the tau protein. So using antibodies that were targeting the N-terminal part of the protein, that facilitated together with the ultrasensitive assays to really capture this N-terminal, which are more abundant in the blood. Again, biochemistry is very important. Here I would add a third important thing in research is that the availability of very good cores. Here actually there is a good example of many people working in longitudinal, very comprehensive characterized cohorts of patients with Alzheimer's or all the continuum of Alzheimer's disease, and this has been also absolutely key in order to develop and validate all these biomarkers. So let's do a review of biomarkers in cognitively impaired individuals. There's a lot of data, but I think it's also to have, in a nutshell, the main data in Alzheimer's disease in people already with symptoms. So that was the first paper, phospho-tau 217 in JAMA Neurology by the Biofinder group. And here it was shortly after the phospho-tau 181, so things were very, very, very quick. And here we can see analysis under the curve to discriminate Alzheimer's disease versus non-Alzheimer's disease, which are extremely, extremely high. This is based with the Lilly phospho-tau 217 assay. MSD-based, extremely good, but obviously it's difficult to translate it into the clinics because it's not an automated or assay, and it's not commercial. Later on, other assays came. This is from ALSPath, semi-automated, based on Simoa, different cohorts, including also the São Paulo cohort, and here with very good also sensitivity and specificity to differentiate Alzheimer's disease versus non-Alzheimer's disease. Or lately, we did a multi-center study using the Lumipulse phospho-tau 217. This is a fully automated biomarker. Most of the clinical routine labs in most of the hospitals can use. So this is not something that only specific labs in the world can do. I would say all hospitals can measure this phospho-tau217 with an automated assay. And here we validated, and this is important, with pre-established cutoffs that we can look at different cohorts and detect Alzheimer's disease with a high discrimination accuracy, even also with primary care centers. Actually, not only this, also it's important, especially in a public health system, the economy. And actually, we did a calculation. You can look at this QR. You will find a shiny app, and you can calculate the savings of using this plasma phospho-tau217 compared to do a lumbar puncture or amyloid PET. All right, so there are plenty now of plasma biomarkers, so things have changed a lot in a very, very, very short time, which is good. Actually, I think that every month there is a new plasma phospho-tau assay. So it's good to do this head-to-head comparisons. This is the one that we did in our hospital, in Hospital del Mar, but there are other publications in other centers. And here what we did is in exactly the same patients to measure different biomarkers and compare the area under the curves, the discrimination accuracy, using as a standard of truth the lumbar puncture, which is what we do routinely as to detect Alzheimer's disease. And the good news here is that we don't have one or two or three, but there are several plasma biomarkers that are very good. If you look at the color code, the green ones are the phospho-tau217, so all of them from different vendors, different companies are extremely good, but there are also some phospho-tau181s that are also great. So we have several plasma biomarkers that are really good. Now the question is, well, which biomarkers do we use? Because we have many that are extremely good. So that's why during the last year, we gathered together different people from the Alzheimer's Association, and we did a systematic review of all the biomarkers that were published at that time. That was until October, but of course from October from now has changed things a lot. But we did this to review all the plasma biomarkers available, and we look whether these biomarkers with the publications fulfill this criteria. For a triaging test, that is to a negative result of the biomarker, rule out the pathology, but if it is positive, you have to do a second test to confirm, lumbar puncture or amyloid PET, or as a confirmatory test. Obviously, the sensitivity, the specificity that you require for this context of use, it is different. For confirmatory tests, we need a sensitivity and a specificity of 90%, while for a triaging test, the specificity can be a little bit lower, 75%, because you can have a second test to confirm. So we'll review all the biomarkers, and these were the winners, let's say, at that time. Probably things will change, and actually we'll meet every year to update these biomarkers list, and probably some of them will now enter this kind of a list, or maybe some of them will disappear if they, with new data, do not accomplish this criteria. But at the moment, that was October, all these tests here fulfill this criteria of triaging test, but only one fulfill the criteria of confirmatory test, which is the mass spec phospho-tau217. I think this will change, and there are now some automated immunoassays that are really good, and probably we can also use it as a confirmatory test. Something that now is generating a lot of discussion is whether to use the phospho-tau217 alone compared to the ratio with a beta 42. And in fact, there was recently the FDA approval of the ratio phospho-tau217 and a beta 42. So here there's a lot of discussion whether to use one or another, something that we can discuss. What is the advantage or disadvantages of the ratio? So the main advantage, I would say, is that sometimes it increases a little bit the accuracy using the ratio, and especially it improves the negative predictive value. And the main thing is that reduces the intermediate zone. So as many of you know, what we recommend when using the plasma biomarkers is to use two cutoff approaches. So if you are higher phospho-tau217, it's a confirmatory, no other test needed. If you are lower to the second cutoff, you can rule out, exclude the disease, but then there is the intermediate zone, the gray zone, in which you need to do something else, an amyloid PET or CSF. Using the ratio reduces these intermediate zones. That's, I would say, the main advantage of using the ratio. However, it has several disadvantages that can be summarized in that A-beta-42 is quite tough from the pre-analytical point of view. It is in CSF, it has been quite complicated when using it in CSF, and it is now also in plasma, and that means introducing more variability in the plasma measurement if you put A-beta-42 in that ratio. Also, the effect sizes between amyloid positive and negative, it's not very big of A-beta-42, and this introduces less robustness, because if there are technical changes, variability, then it's easier that there are no differences between the two groups that you are comparing. So introducing A-beta-42 may reduce the robustness of the assay with the ratio. So that's something to discuss, but this, in my opinion, is that probably using also phospho-tau217 alone, it's better than using the ratio. All right. Let's go to preclinical Alzheimer's disease in cognitively normal, which is the specialty in BBRC. We have the alpha cohort, which is focused to preclinical Alzheimer's disease. And it was 3 years ago, so not a long time, in which we published this paper, Marc, former PhD student in my lab, in which we model different changes of different plasma biomarkers in the continuum of Alzheimer's disease. In the horizontal axis, you have the amyloid PET centiloids, and we see how many of these biomarkers change. And we see that some of the phospho-taus, 217, 231, are those that increase earlier with subtle changes of amyloid PET. That was not so long ago, 3 years ago. That was confirmed in the Biofinder cohort by Nick Ashton. Again, if you look here at this 231, so this is the quartiles of centiloids, and 231 really increases very early in the first quartile, while others like 217 increases later and then keep up increasing. So it's more like a staging biomarker, while 231 it does a plateau later on. So that was the analysis, the ROC analysis that we did at that time. And as you can see, the areas under the curve are fine, but are not great, 0.7 something, that at that time we were quite happy. I mean, we were talking about a plasma biomarker in preclinical Alzheimer's disease, so this is quite great. It was just 3 years ago. But things have evolved so quick that that's not enough. And actually now we see studies, this is from Biofinder, cognitively normal, using mass spec, and you see area under the curves that are extremely, extremely high. This is mass spec, so not very useful for the real world, but now we have some immunoassays. So this is from Armand González Escalante, PhD student in my lab, using automatic NTK assay and phospho-tau217, not in people with symptoms, but in preclinical Alzheimer's disease, has very high area under the curves, and this is an automatic assay that can be also used in most of the labs. So in a very short time, things have changed really dramatically. Not only this, but we need biomarkers to detect the pathology, to detect Alzheimer's disease, but more importantly, also to detect who will progress to cognitive impairment. And in the alpha study, in BBRC, we have the fantastic alpha cohort, in which now we have been following our participants for more than 10 years. That gave us the opportunity to measure the plasma biomarkers at baseline, that was in 20 in blood samples that were kept from 2013, and we have longitudinal data, cognitive with the PAC composite, of cognition for more than 10 years. And we tested whether the baseline blood biomarkers are associated with cognitive changes 10 years later. And what we see is, this is also, again, from Armand, that here you can see it. So this is different biomarkers. This is the beta. So if it is negative, higher levels of these biomarkers are associated with worse cognition at follow-up, a longitudinal follow-up. And as you can see, many of these phospho-taus, not only 217, but 181, 231, and the ratios are associated with cognitive changes. So these biomarkers not only detect amyloid deposition in asymptomatic people, and symptomatic as we have seen before, but also are associated with cognitive changes at follow-up. So another area of research, which is, I think, extremely interesting, is subjective cognitive decline. This is probably that here there are many neurologists, many clinicians, and this is a group of patients that are quite frequent in the clinics, and I believe that we do not care of we don't provide good enough care because most of them, we just see these patients, we follow up for some years and see where they progress. So also in BBRC, we have a core that is focused on subjective cognitive decline, which we call it beta-ARC, Alzheimer at risk, 200 participants, and Federico and Jose analyzed several plasma biomarkers in collaboration with Gothenburg, and also some of them also measured in our lab. And again, we will not go through to all these biomarkers, but the good news here is that we have several biomarkers that in these early symptomatic individuals, they also detect amyloid pathology as compared with lumbar puncture. However, here, I would like to highlight something which is key in order to understand how we use blood biomarkers. It's not only about diet under the curve, it's not only about the sensitivity and specificity, but blood biomarkers and any biomarkers have to be interpreted always in their clinical context. And the clinical context in subjective cognitive decline is that the pretest probability, the prevalence of amyloid pathology is lower. And this needs to be understood to interpret the predictive values. So although the R under the curves and the sensitivity specificity, which depend on the test itself, is very good also of these plasma biomarkers in subjective cognitive decline, the pretest probability is lower because there are not so many Alzheimer's patients in subjective cognitive decline compared to an amnestic typical case in MCI. So we calculate the predictive values. The negative predictive values are very high. A negative result of plasma biomarker can really rule out, exclude Alzheimer's pathology, but the positive predictive values are not high. So maybe in subjective cognitive decline, which is there's quite a lot of discussion whether we should use biomarkers or not, maybe we could use biomarkers to exclude pathology, but not to confirm pathology. That's, again, the importance of interpreting the pretest probability. So phosphotau, there are many of them, 205, 212, 217, 181, 243, different variants, different truncations, that it's becoming all of a mess. So let's go again to the biochemistry. So this is tau that we will talk about this morning. Is actually the 2 and 4 repeat, because as you know, there are 4 repeat to repeat, but here's the full length tau. Here are the phosphorylations, and also the different phosphorylations, also some truncated forms. I will not go now into details about the biochemistry, but I want to also highlight a couple of things here. First of all, different phosphorylations can tell us different information. So for practical terms, the 217 is great as a diagnostic tool for the clinical use. But as we have seen, the 231 increases very early in the disease. And this is consistent with these findings of Gemma Salvador, at that time a biofinder. Now she's back with us, showing that in the pathology, 231, it is here, is more related to, in blue, amyloid plaques than tangles, while the 2-1s indicating that it's a very early biomarker of subtle changes. While 217, it's more both, and 181, again, it's a tangle, it reflects more a bit tau pathology. So different phosphotaus are reflecting different mechanisms that occur in Alzheimer's disease. Anyway, all these phosphotaus are very early, and in fact, something that I think is very good that has done, the new criteria of Alzheimer's disease, is to separate the T of the ATN with T1 referring to the soluble phosphotaus, that includes 181, 217, 231, and T2 as an indicator of insoluble tau, the tangles. And there are other fluid biomarkers that better indicate the insoluble tau, the tangles, like, for example, the MTBR243. There was a lot of discussion of whether the phosphotaus are more indicator of the A of the ATN or the T of ATN. But this is, again, only a terminology and nomenclature discussion, in my opinion, which is quite common in our field, having terminology discussions. But the point here is that these phosphotaus increase very, very early and are indicating of this, I would say, tau-related changes, but very close to amyloid deposition. So we have different taus, different forms that are reflecting different things that happen during the continuum of Alzheimer's disease. Another thing that I would like to highlight is that, and here again, the biochemistry is very important, is that we have different taus and different fragments, and that was Fernando González Ortiz in Gothenburg's lab that described that there is a brain-derived tau, but there is also a big tau, a peripheric tau, that has this exon here that is and this big tau is more frequent in the periphery. So of course you can use different antibodies to detect specifically the brain-derived tau, which is specific from the central nervous system, or to detect the peripheral big tau. Actually, there are some of the assays that we have been using for many years that we didn't know that were more specific for central or peripheral tau. Just as I show you, for example, in ALS, there is an increase in some assays of phospho-tau 217, which is counterintuitive because of course, ALS, it's not Alzheimer's. But this happens because there is an increase of the big tau. And this happens with the ALSPath assay, but it does not happen with the Lilly assay. Okay, I go quickly with that because I think I don't have much time, but just I would like to highlight two more points that phospho-tau 217 and some of the assays have been starting to be used in the clinical trials, and we see clear results. But the thing that these plasma biomarkers change with clinical trials, this doesn't mean, and this is a discussion, that we can yet use it as a surrogate marker. And here I recommend you to use to read these two articles discussing whether we could use amyloid PET as a surrogate marker or phospho-tau 217. I think that we are not there. There's a good correlation with phospho-tau 217 or many biomarkers with amyloid PET, the standard of truth. But at individual level, the correlation is not so good. But maybe at some point we can use phospho-tau 217 as a surrogate marker as it is used in HIV, the viral load. That would accelerate a lot the biomarkers. Another thing that also, because I think it's quite interesting, what is the meaning of tau? Because we think in tau in Alzheimer's disease, we have seen that sometimes also increases in ALS, but there are some funny things I would say like this. For example, phospho-tau 217 is increased in newborns. That's something that we found some years ago. At that time, we were measuring, investigating umbilical blood from newborns. For other purposes, we were looking for rejuvenating blood factors, but we measured the blood biomarkers. And interestingly, the phospho-tau 217 is also increased in newborns. We don't know the reason, but we think that as there is synaptic pruning at that time, maybe there is this increase of phospho-tau. So phospho-tau probably is reflecting from the physiological point of view other very interesting things that are beyond Alzheimer's disease that also merit attention and more research. So in summary, I'll try to explain that we can measure now tau because of the ultrasensitive assays, the good course, and the importance again of knowing the biochemistry. Phospho-tau 217 probably the best for cognitively impaired, but we can also in cognitively impaired individuals measure now reliably many biomarkers, not only with mass spec, but also with some immunoassays, and especially 217 and 231 at the very early stages are increased. We can also use plasma biomarkers in subjective cognitive decline, but it's always a key to understand the clinical context, the test probability, the prevalence of the disease. And also we have seen that different tau variants are reflecting, it's not all the same, are reflecting different pathophysiological mechanisms that are occurring in Alzheimer's disease. And maybe we can start also using plasma biomarkers in clinical trials that I think will accelerate everything a lot. And with that, I finish also thanking Fundación CIEN for inviting me and also all my group, which is here. Thank you very much.
Thank you, Marc, because in your summary, you start for a huge questions for the audience. Thank you very much.
Thank you.
Albert?
So good morning. Well, first, I would like to thank the Fundación CIEN, Pasqual, and Fundación Reina Sofía, and also Mercè for the introduction. So today I will talk about a little bit similar topic, but I will go inside the brain. So what happens with tau inside the brain? These are my disclosures, but none is relevant for today's talk. So synapses are really very, I think, exciting structures of the brain, and as you know, they are the basis of our cognitive function and behavior. But they have one challenge, is that very small structures, very dynamic and very complex, and they're very difficult to study, especially in human brain. As a clinician, I'm more interested in the loss of synapses, and we know from the literature that the loss of synapses occur in several physiological and also pathological conditions. For example, normal aging, this is a study in monkeys where they show that as they age, the monkeys in the brain tend to lose synapses, especially in the frontal cortex, and this may underlie some of the cognitive changes that they experience when they age. Also, loss of synapses underlie many pathological processes in brain disorders. For example, these are in major depressive disorders. It has been shown that there is a loss of synapses in the prefrontal cortex. Here, using electron microscopy, which is the standard technique, has been the standard imaging technique to study synapses in the brain. And also, there are similar data in schizophrenia. So the major mental diseases, they have a loss of synapses in the brain. But of course, I'm more a clinician, and I'm dedicated to Alzheimer's disease. And this is a very nice paper, not very old, from the 2017, but they use a very classical technique called GGOX to show that in Alzheimer's disease, there is a prominent loss of spines in the brain. As you can see here at the bottom, you can see there is a clear loss of spines compared to controls. And what is interesting is that they also look at people with brain pathology, so Alzheimer's disease pathology, but without cognitive changes, so we call preclinical AD. And you can see that they still have some spines conserved compared to people with Alzheimer's disease. Especially the stubby spines are conserved in preclinical AD. So that's important because that means that dendritic plasticity is probably one of the mechanisms that underlie cognitive resilience, and I'm sure we will hear much more in the next talks. So also, there is a classical paper that in Alzheimer's disease, the synapse loss is the major correlate of cognitive impairment in Alzheimer's disease. This is a classical study in the 90s by Terry. Here they use very classical and traditional scores. And other modern, more recent papers, also this is in the MAC cohort, they also show that in Alzheimer's disease there is a 30% decrease in synaptophysin, which is a classical marker of synaptic marker. And also many of the proteins such as BAM2, SNAP25 were associated with cognitive function. And interestingly, for each unit of synaptic protein, there was a less risk of cognitive decline. So I think that builds on the fact that synapses are at the center of cognitive resilience. So, however, in human brains, synapses are extremely difficult to study. And being in Spain, I think it's nice to quote Santiago Ramón y Cajal. He got very frustrated when he looked at the stainings because he said that, well, it looked like a jungle. It was very difficult to decipher what was going on, especially in each cut. And they say it's a jungle that appears impenetrable and indefinable. So it has been a major limitation, a technical limitation, how to study synapses, especially in human brain. So we were lucky to some years ago, to hear about this technique, ray tomography. This was developed at Stanford University by Micheva, and they had a very clever idea. This is a technique, a microscopy technique, which is based on a very simple principle. If you have a very small structure, but if you cut the samples with ultra-thin sections, very, very thin, you will have the presynaptic compartment in one section and then the postsynaptic compartment in another section. So then you can stain with different markers and then you can reconstruct the images. So by cutting the sample, like a physical method, you can get an enhanced resolution. So then we in our lab, we implemented this technique 10 years ago. And we have been applying to answer different questions related to the human brain and human and neurodegenerative diseases. This is a summary of the technique. It's very tedious. There is one major limitation that you need fresh tissue. So that means that you have to go to the brain bank immediately with each brain donation. And of course, that limits the availability of tissue, and in fact, this is only implemented in very few centers in the world, this technique, because it's very tedious, it's complex. You need to get the fresh sample, and then you cut it, and then you embed it in a special synthetic resin so that you can cut it in very, very thin slices of 70 nanometer sections, and then you stain it with different markers, pre- and post-synaptic markers, and then you reconstruct the images using fluorescence microscopy. So that takes time. But then once you have enough, we have around 50 cases right now in our biobank, we're using these methods, and then you can answer very, very key questions. Here you have an image of what we get. This is a synaptogram. This is in red presynaptic marker, synaptophysin's postsynaptic marker, PSD 95 in blue. And you can see here that you can have a very nice image of a single synapses in human brain. This is always human brain. So you get the very nice images first, and you can see here with different sections, which we call a synaptogram, so you can have a very nice idea of how the synapses look like. And with this, we have been addressing different questions. This is a paper that we published with Martí Colom, who was one of my PhD students. And in here, we demonstrated for the first time that synuclein is inside synapses, aggregated synuclein is inside synapses in dementia with Lewy bodies. Here, you can see here that besides Lewy bodies and Lewy neurites, you can see small aggregates inside the synapses. And this is, we think this is important because it can contribute to the synaptic dysfunction. This is one of the central mechanisms in dementia with Lewy bodies. We also apply these techniques to amyloid beta because besides synapses, you can get enhanced resolutions. And here, what we saw is that we could image an entire amyloid plaque. You can see this is an amyloid plaque. It's more than 400 sections to get an image, and then you can characterize the different areas of the plaque using this technique. And also, this is another example that we published. Like this is the first case of a patient in a clinical trial. This was a patient treated with a BACE inhibitor, berubecestat, who died. And then we look at A-beta in the brain of this patient, and there was no major changes in total A-beta, but when we look at synaptic associated A-beta, then we could see that there was less synaptic associated A-beta compared to untreated subjects. So that's a tool that you can have, you can get different answers. And going to tau, because the talk is about tau, I would like to show these results that we did in collaboration with Tara Spire-Jones in Edinburgh, in which we investigated oligomeric tau in synapses in human brain. This is a collaboration with Martí Colom, who moved to Edinburgh to do this study, and also Sonia and Erica in our group. And here we examine a cohort of brains from Alzheimer's disease cases and controls, and two different regions. And what we show is we found that, of course, synapses densities were reduced in Alzheimer's disease cases. This has already been demonstrated. But that we took advantage of array tomography to see what happened at the synapses with tau. And one interesting thing is that what we found that in areas with low tau pathology, with low neurofibrillary tangle pathology, there was already oligomeric tau at the synapses that can be detected. So in areas, very early deposition or accumulation of oligomeric tau in areas without prominent tau pathology. And also we look what happened with other tau isoforms. We look at misfolded tau or phosphorylated tau, and what we found is that oligomeric tau using T2 antibody was the most prominent species in the synapses. So it seems to be a very early accumulation of oligomeric tau in these structures. Also, the beauty of array tomography is that you can dissect what happens in the pre-synapses compared to the post-synapses. So you can get a sense of how tau is distributed across the synapses. And here what we found is what's very interesting. There was a gradient from the pre-synaptic tau to post-synaptic tau. So there was a more oligomeric tau in the pre-synapses and then less in the post-synapses. So there was a gradient from the pre to post, which could be interpreted as an indirect evidence of tau transmission across the synapses. Of course, this has to be very cautious because it's a cross-section, a neuropathology is a cross-section, so you always have to be careful. But it looks like there is a trend, there is a gradient from pre to post. And also, in our group, we have array tomography implemented, but as I said, this is based on the physical section. But then we ask, what happens if we combine array tomography with a super resolution microscopy so that we can even get more spatial resolution to see what's going on in this small structure. So we chose a STORM. STORM is a super resolution microscopy method that is available in Barcelona, led by Silvia Pujals. And this, I won't go into details, but basically using a laser, you basically pushes to the fluorophores to the dark states, and then they go back to the excited states very much by randomly, and by repeating this very fast, you can measure these photons and get an enhanced image, an enhanced lateral resolution. So this is an example. This is how it would be one array tomography images. You can see the images that we get with array tomography pre or post, but when you apply this sample to a STORM, then you get a much higher resolution. You can see clearly the pre and the post synapses very clear in a much more defined way. So this is another study, this publication, more technical publication led by Erika in our group. And here you can see it's very nice. You can see clearly even the synaptic cleft here. Here between the pre and post synaptic. So this is really nice. The resolution is very time consuming. It takes hours to get just one single image, but you get, and also the size of the synaptic clefts here, it's really pretty consistent with what has been described by other techniques. So going back to tau, so we asked, can we, with this enhanced resolution, can we detect tau, oligomeric tau, inside the synapses? And the response is yes. We were able to see both oligomeric tau here, inside the synapses, as you can see in white, and also using 88, which is a classical tau marker for more pathological tau. We also see that tau is inside the synapses, and we can be detected with this technique. So we think this is important because oligomeric tau has been quite elusive in the synapses, and we believe, we propose that oligomeric tau plays a role in the spreading from pre to post synapses, likely contributed to the extension of the pathology in Alzheimer's disease. And this is supported by other studies. I won't go into details. This is a study led by Oscar Hansson based on amyloid PET, tau PET, and they analyzed the patterns of tau PET across the cohort, and the model predicts that the model of tau spread matches pretty well with the connectivity patterns, so suggesting that the pathology spreads through functionally connected neurons in Alzheimer's disease. So I think it's compatible with the idea that perhaps the oligomeric tau is playing a role here in this model. So one word about anti-tau therapies. Tau therapies are really lying behind amyloid therapies. There's less research. Tau is a more complex molecule. There have been less clinical trials, and there is an intracellular target, and there is less tau. So there are some complexities in anti-tau therapies, but there have been two very good news, I think, in the field. The one was the study from Biogen, the BIP80, which is based on an intrathecal antisense oligonucleotide against tau that lowers basically the expression of tau. And they reported and they have published that injection of the intrathecal azoles can lower the tau pathology in the brain measured with tau PET. Here you can see 2 patients in placebo. There is a tendency to increase over time, but the treated patients, there is a tendency to reduce the amount of tau in the brain. And I think this is extremely surprising, extremely exciting, because it tells us that this is the first hit with a therapy on tau pathology, and that means that we can reduce tau pathology in the brain. And to me, that's important because it means that neurofibrillary tangle pathology are much more dynamic lesions than we thought. The turnover of tau here because we are just reducing the production of tau. It's probably the turnover of tau molecules inside neurons is very high. So that's a very good thing, I think, very promising for the tau field. And Biogen is advancing to a phase 3 program probably in the next months or year. And what happens with antibodies? Antibodies have been much more challenging. All these are a list of antibodies that have been negative, all have been discontinued, all have shown no efficacy. With just one exception, and I will talk about bepranemab, which is an antibody directed against the Mead region epitope that they reported results. It was negative because the primary outcome was negative. Here you have a summary. The primary outcome was the CDR sum of boxes. It was negative, so it has to be considered a negative study. But they show that there was a reduced rate of tau accumulation in the brain in some regions by 30–50%. And then some of the secondary endpoints, like ADAS-Cog, there was a slow decline in around 25% in the ADAS-Cog. So I think there's some hint, there's some signal here. We need to see how this goes, if they move and they repeat another trial. And also there was an effect in 2 predefined groups, like with low tau burden and non-APOE4 carriers. So there is something here. I personally think that probably the brain shuttles will have a role here in the future with this tau, because tau is an intracellular target. So it could be that brain shuttles or antibody transport vehicle, which is the technical name, will have a role in the future trial designs. So, and then just to finish, neuropathology is pretty much a descriptive discipline. So we wanted to see how tau behaves biologically. And then we set up a model with Sonia, Erica, and Aleida. In our lab, we set up a cell culture model to measure the bioactivity of tau, or tau seeding activity, using a cell model, which is this tau FRET biosensor cells. This is a cell model developed in Marc Diamond Labs, and is commercially available, so you can buy the cells. And the principle is that this cell line expresses tau, mutant tau, fused to 2 different fluorophores, CFP of YFP. And when you add pathological exogenous tau, then the cells tend to aggregate, the tau, endogenous tau tend to aggregate, and then emits fluorescence. It makes FRETs. So and this can be measured by fluorescence, by imaging, or by flow cytometry. And here, this is the principle. So you can see if you treat the cells with a brain extract from a control, you don't see fluorescence. But if you treat with a extract from Alzheimer patient, then you see some fluorescence. And here you can see some examples, for example, from our cases, controls or Down syndromes without prominent pathology. You see no FRET or minimal FRET when we treat with samples from sporadic AD, patients with mutations, or the Down syndrome with Alzheimer's disease, you see prominent FRET, which means which is a reflection of tau seeding. And I think this is important because it's not how much tau do you have in your brain, but how bioactive really is your tau, the tau that you have. And then just to finish, Erica has been performed different experiments with different cases. And then going back to the samples from the Neuron paper, she showed that oligomeric tau correlates with tau seeding activity in sporadic Alzheimer's disease. So it's the best correlate of tau seeding is oligomeric tau, which also reinforces the role of oligomeric tau in Alzheimer's disease. And you can see the details. She will be presented the poster today in the poster 16. Just to finish, to summarize, I think we believe that synaptic tau pathology is a key event in the pathogenesis of Alzheimer's disease. It's important to develop techniques such as array tomography or super resolution microscopy to study synapses in human brain. Also, we have shown evidence that oligomeric tau is the predominant tau species in synapses in Alzheimer's disease and seems to play an important role in determining the tau seeding activity in the disease. And I think these data are important when developing anti-tau therapies in the future. So thank you very much. Yeah.
Thank you too much. because that is a recording of the effects of the tau, and introduce the two news speakers. Please.
Thank you very much. Pasqual, thank you very much for the invitation here. It's a wonderful venue. I'm not sure how much drama I'm going to provide for you on this stage, but it's appreciated nevertheless. So I'm a clinical neurologist from London. I lead the UK's National Prion Clinic. So what I'm going to talk about is a little different from some of the other talks here, so I ask that you bear with me for a little while, because when we get towards the end of the talk, it does have a kind of tau finale. But what I'm hoping to persuade you this morning is by studying this kind of rare disease that is seen as somewhat an unusual and perhaps sinister relative of other neurodegenerative diseases, maybe we can learn something about fundamental mechanisms. So initially, I'm just going to provide a little bit of background about prion diseases, what they are. Okay. So prion diseases are transmissible disorders of humans and animals, and they're fatal in all cases. And they're quite well known, or historically they've been well known for animal diseases that are endemic, like sheep scrapie. And perhaps one of the most famous exports from the United Kingdom to the rest of the world was mad cow disease, or BSE, examples of which you can see on the bottom half. These diseases cause cognitive problems for sure. Typically in humans, a kind of multifocal, rapidly progressive dementia with impact on executive function, working memory, but pervasive cognitive decline, but also ataxia, myoclonus, and motor signs as well. It's really a catastrophic neurodegeneration. And in the brain, you see spongiform change in gray matter and deposits of abnormal prion protein. Now, importantly, the disease was demonstrated to be transmissible by inoculation of brain material into unaffected animals, by accidental experiments done in 1936. And then Colton Gajdusek and Michael Alpers, Colton Gajdusek subsequently went on to win the Nobel Prize for this work, showed that human diseases like Kuru and subsequently CJD were transmissible by inoculation into chimpanzee. And later work established very effective transmission models in mouse. Just some background about the epidemiology of the human disease. This data from the epidemiology unit based in Edinburgh in Scotland. So in red, you see the total number of human prion disease cases that we see in the UK. And these cases are the basis for the discovery that we made relevant to Alzheimer's disease. So we see in the UK around 130 to 150 cases of prion disease in total in a typical year, which works out to around 2 per million of the population. The majority of these are sporadic CJD, so just similar to Alzheimer's disease. It's a sporadically occurring disease of middle-aged and elderly people who typically survive for around 5 months with this condition, and it's completely untreatable. And most of the remainder of those 140 or so cases have an autosomal dominant form of inherited prion disease, where mutations are found in the gene that encodes prion protein PRNP on chromosome 20. So what you can see at the bottom of this chart are the acquired or transmitted forms that we see, and in orange, variant CJD. Now, variant CJD was the human form of mad cow disease, human form of BSE, transmitted in the diet largely to the UK population, but 14 other countries. France was perhaps the worst, and there were some Spanish cases as well, but vast majority of cases were in the UK. That peaked in 2000. And has subsequently been in decline. The last case of variant CJD we saw in 2016. An expectation from modeling is that we will most likely see no further cases. There is the possibility that we will see a small number, but most likely no others. But there are other forms of transmitted prion disease too, called iatrogenic CJD. Most of these in the UK were the result of a treatment of children with short stature, with human growth hormone that was purified from pituitary glands. And this was a program that was developed in the late 50s, treatment started in the 60s, but ramped up in the 70s and early 80s until synthetic forms of human growth hormone became available in 1985. Now, we've seen 83 cases in total, so this were children treated with these products. Then after many years, even up to 40 or 50 years later, will develop an iatrogenic form of CJD. But these are spread over many, many years. So as you can see in recent years, the recent, say, 10 years, we've seen about 6 cases of iatrogenic CJD. So it's not a frequent condition at all. So when talking about similarities between prion and other neurodegenerative diseases, it's interesting to note the progress that's been made in determining the structure of the abnormal proteins that are found in brain tissues. And when you look from this perspective, there are striking similarities across the board, because cryo-EM shows that essentially these are like amyloid-type fibrils formed of PRP. These are rodent prions. We don't yet have structures of human prions, partly because of the infection control issues, but rodent prions are formed of parallel in-register beta sheet forms of normal prion proteins. So there's a major transition in structure, just like you would see for amyloid beta, for tau, for synuclein, and others, TDP43. There's a major transition in structure from normal, in PRP's case, a self-surface GPI-anchored, largely alpha helical protein into one that's predominantly beta sheet. Okay, so that's your background about prion diseases. So where does amyloid beta come in? Where is this story? Well, actually, it's a really long story. So Colton Kajusek, I mentioned, that transmitted animal and human prion diseases to chimpanzee and then subsequently other species. He also tried to transmit other neurodegenerative diseases. But the results of these experiments are a little patchy and unclear, and really, I think the first definitive progress was made by Ros Ridley and Harry Baker in the 1990s, who injected Alzheimer's disease brain material into monkeys, marmosets in particular, and they demonstrated that you can get amyloid beta pathology appearing in these monkeys after a long incubation time. There was no clinical symptoms here, but this was the first demonstration of experimental transmission of amyloid beta in these animals. But subsequently, this, I think, was taken much further forward, major steps forward by Larry Walker and Matthias Jucker, who did similar type of experiments in transgenic mice that express a mutant, sometimes overexpress mutant human form of amyloid beta, demonstrating that you can dramatically bring forward the appearance of amyloid beta pathology by these inoculations. But the major question outstanding from this is, well, that's an interesting phenomenon, and amyloids, it's not that surprising that amyloids might behave like this, but is this really relevant to humans? Is it really relevant to patients? And here was the discovery moment from our clinic. We were, every, say, 3 months or so, the clinical team, we have a joint pathology meeting with our neuropathologist, Professor Brandner and Azon Mukhtani at the multi-headed microscope. And on this occasion, we decided to review all the cases of iatrogenic CJD that we'd seen in recent years. And one thing that was really striking that came out of that was, whilst we could see the full pathology of iatrogenic CJD, the majority of these cases who were treated as children but were now aged somewhere between the mid-30s and 50, so very young for neurodegenerative diseases, they all had a really marked amyloid beta cerebral amyloid angiopathy that was completely unexpected. So we wrote up those findings in Nature 2015, and remember, these were people that had died of iatrogenic CJD. They didn't have clinical manifestations of amyloid beta cerebral amyloid angiopathy, but nevertheless, it was a striking and unexpected finding. And that triggered people to look in their own archives, their own pathology material, particularly for people not only that had been treated with cadaveric human growth hormone, but also those that had received contaminated dura mater, which is the major other cause of iatrogenic CJD. And there were then tens of additional reports that appeared in the literature over coming years. And currently in the latest database, there are more than 100 cases now of iatrogenic amyloid beta cerebral amyloid angiopathy with very early ages of onset, sometimes diagnosed in life, sometimes post-mortem, without obvious genetic cause, all of whom are childhood procedures that involve either cadaveric dura mater or human growth hormone or other exposures that might be relevant to prion disease. Okay, so this is established now. So many of these papers were published 5 years ago or so, but if amyloid beta as a proteinopathy is transmissible, I think a key question also then is, well, what about Alzheimer's disease? Alzheimer's disease is the major disorder associated with abnormalities of amyloid beta. Would it be possible that over time, after having cadaveric human growth hormone injections that we now know contain abnormal amyloid beta and can transmit the disease to transgenic mice, could that ultimately in humans lead to the full clinical picture and pathological picture of iatrogenic Alzheimer's disease? Well, of course, as you know, Alzheimer's disease is a clinical syndrome, and as an Alzheimer's disease dementia associated with loss of everyday functions, it's also heterogeneous with predominantly amnestic presentations, but also frontal, visual, and language forms that are well recognized, characterized in the brain by amyloid beta deposition in the parenchyma and blood vessels as CAA, and then, of course, neurofibrillary tangles of hyperphosphorylated tau. And there's compelling evidence that amyloid beta is the root cause of that. In other words, there is a cascade where abnormalities of amyloid beta appear first and then trigger tau pathology to become abnormal. So there's an established case about the human transmission of amyloid beta pathology, but then what about Alzheimer's disease? So subsequent to the publicity around the original paper from 2015, we started then to get referrals from clinicians around the UK. Now, I want to just be clear, we don't know the names, the addresses, the dates of birth of the 2,000 or so children that were treated with cadaveric human growth hormone. So everybody we see has to have a reason to be referred to us, either because they've gone to the doctor and said, I had this treatment, I want to be checked out, or because they've got symptoms and a doctor appreciated that there might be a connection between the two. So this is not a cohort study. This is not a formal epidemiological study of everybody that was treated in this way. It's just a report of the people that were sent to us. In fact, all of the people that were sent to us by their doctors that have been treated by with cadaveric human growth hormone. So we had 8 referrals between 2017 and 2022, and none of these people subsequently developed CJD. So these are treated people, but they haven't got CJD. So as I said, 83 cases of iatrogenic CJD out of around 2,000 treated. So the attack rate of CJD is still very low in this group. So 5 of them had an early onset dementia, 2 with an amnestic presentation, 3 non-amnestic. A couple of those were language, 1 was dysexecutive. And these are heterogeneous cases, okay? And I'm not going to go through the details of each one, but suffice it to say that 3 of them had already had an established diagnosis of Alzheimer's disease. And in other cases, there was either biomarker or imaging evidence of Alzheimer's disease. It would be wonderful if we saw these people from around the UK at early stages, could engage them with research and do a full panel of clinical assessments longitudinally and biomarker assessments, but it wasn't like that. Many of these cases were referred to as in a very advanced state where it wasn't really appropriate to do follow-up investigations like that. There was a further patient that had MCI who died of an incidental cause and was found to have amyloid beta pathology at post-mortem and some tau pathology. And there was 1 asymptomatic and 1 person with subjective symptoms. The asymptomatic individual had some abnormal biomarker results, and the subjective symptoms individual had a single microbleed. So how had these people been treated? Well, interestingly enough, there were a number of different preparations of cadaveric human growth hormone that were used from the 60s through to 1985. But in the UK, at least, all of the cases of CJD have been associated with 1 particular preparation called Hartree-Wilhelmi, or HWP. And one thing that we noted is that all of the patients that had a diagnosis of Alzheimer's disease or early onset dementia had received these batches. And there was nobody that had received none of the Hartree-Wilhelmi preparation, which is an identical feature to the iatrogenic CJD cases. We also wanted to look for alternative causes of this. As I mentioned, they're a very heterogeneous group, so we did gene panel testing where we can, but we didn't find any genetic explanations in the group. So as I mentioned, all of the patients were treated with HWP, and this was a type of cadaveric human growth hormone that previously we'd shown, because the vials of this material had been stored at Porton Down, which is a kind of military base in Wiltshire. They'd been stored for legal reasons for 30 or 40 years, and we managed to get hold of some of these vials and demonstrate that they do contain abnormal amyloid beta, and that if you then solubilize that material, inject it into transgenic mice, they will report seeding of amyloid beta after those injections. So the people that developed early onset dementia had received preparations that we know seed experimentally. So whilst we looked for other causes, the only thing in common was this seeding material, which is what led us to the hypothesis that this was actually a transmissible form of Alzheimer's disease. So just in a little bit more detail, this was one case, case 3, who had high-resolution T1-weighted MR imaging showing temporal atrophy, and particularly in the temporal lobes, and also had axial PET images demonstrating increased tracer uptake in the cortex of fluorbetapan, indicating a high likelihood of parenchymal amyloid beta. And the post-mortem case 1 shows really frequent parenchymal deposits of amyloid beta in the cortex, in the deep nuclei, and also in the cerebrum and some in the cerebellum, particularly cerebral amyloid angiopathy, also parenchymal A-beta, but in one particular area in the insular cortex, there was a dense network of neuropil threads, pretangles, and occasional tangles, and neuritic plaques. So we looked quite hard to try and disprove this hypothesis. Was there some other explanation that could connect all of these cases? I mentioned genetic susceptibility, but what about the pre-existing reasons that they needed cadaveric human growth hormone treatment anyway? Many of these are just idiopathic growth hormone deficiency, but there's a range of different complex disorders. But we couldn't connect together the cases in that way. There was also questions about whether growth hormone deficiency itself might be a risk factor for Alzheimer's disease, but the evidence for that is very limited. And some of the patients had cranial irradiation. In fact, half of them had. But there's really very limited data to suggest that there are long-term effects on amyloid beta following irradiation. So we wrote these cases up as a follow-up paper to the Nature paper in Nature Medicine in 2014, and yeah, it really generated quite a lot of interest. It had a massive altimetric score, and there was a lot of debate about these findings on social media. And unsurprisingly, we had a lot of inquiries that resulted from this paper, some of which were for reassurance purposes, and we set up a helpline to make sure we could reassure people. But we also have had quite a number of referrals, and there are some very interesting cases coming out of these new referrals. So you can see this publicity is helping us get access to the wider cohort that for privacy reasons, we've hitherto had no access to. So I just want to tell you briefly about, coming to the tau denouement, that what we've found in some of these more recent cases. So since the Nature Medicine paper last year, we've had 15 referrals of people that were likely to have been treated with cadaveric human growth hormone. 6 were definitely HWP. We've checked, cross-checked those individuals in the database. Some were confirmed to have cadaveric human growth hormone, but not HWP, and then some either for batch coding or for other reasons. We can't yet confirm for sure exactly what material they've had. But we're seeing a range of people now that have had either cognitive symptoms or gait changes from all of these different groups. But the one in particular has come to autopsy, and I think has some really striking findings. Because I think if there was some pushback against the original Nature Medicine paper, it was, well, it's all very well that you've got people with some language disorders and there's a lot of amyloid beta pathology, but really Alzheimer's disease is amyloid and tau. And whilst there was one patch in the insular cortex of one of the autopsy cases, you've not really demonstrated the full picture of Alzheimer's disease in this series. And can you really say iatrogenic Alzheimer's disease without a picture like that? So let me show you this case. So symptom onset in 47, strikingly young. Okay, so normally you're just seeing people with gene mutations at this age. This is not the age where you expect to see sporadic Alzheimer's disease. This case had a lobar intracerebral hemorrhage, age 50, but had a very good recovery from that, but a significant progression of the cognitive symptoms at age 54, and was seen by a cognitive neurologist on the south coast of England, and was diagnosed with semantic variant of PPA. Language involvement progressed. There were navigational difficulties and dyspraxia. By the time we saw the case, like I illustrated with the earlier cases, very severe, bedbound, fully dependent, and speech was limited only to a few words, and the patient died about a year later. No genetics causes found here. Incidentally, the imaging there showing the very severe temporal atrophy and hippocampal atrophy. So we did an autopsy, and indeed there was an acute hemorrhage in the left hemisphere with widespread subarachnoid hemorrhage, but that's not the main thing I wanted to show you. I wanted to show you the amyloid beta and tau pathology. So amyloid beta pathology was really severe throughout the cortex and subcortical nuclei, including the cerebellar cortex. It's a very severe cerebral amyloid angiopathy, parenchymal A-beta of highest pathological grades. But what was striking and was new in this case, compared with the previous cases that we'd reported, was kind of end-stage Alzheimer's disease tau-type pathology that was very widespread throughout the hippocampus, the frontal cortex, occipital cortex, graded stage 6 by our neuropathologists. So essentially, Sebastian Brandner and Zane, our neuropathology colleagues, graded this as the very highest levels of Alzheimer's disease, neuropathological change in this case. So we're currently following up these other new referrals that have come in since the 2024 paper, but I'll wrap up with the kind of formulation and some of the interpretation of these data here. So I think what I've demonstrated is that you can transmit in human amyloid beta pathology in cadaveric human growth hormone recipients. So in particular, this manifests sometimes as asymptomatic cerebral amyloid angiopathy, sometimes as symptomatic cerebral amyloid angiopathy in terms of brain hemorrhages and strokes, but also as a neurodegenerative dementia picture that's certainly within the spectrum of Alzheimer's disease and can justify the term iatrogenic Alzheimer's disease. Even if an amnestic syndrome is not typical, it's more language presentations. So I think these confirm that prion-like seeding and propagation of amyloid beta is not just an experimental phenomenon, it's actually something that happens in humans too, and therefore is an expansion of the prion concept beyond classical prion diseases. And that then questions whether some of the control measures that we use for prion diseases are also relevant to the wider spectrum of neurodegenerative diseases in healthcare settings and in laboratories. And I think that's something that is worthy of further discussion, although I do emphasize the word proportionate there. I think the findings fundamentally strengthen the amyloid hypothesis because we show that there's amyloid beta seeds in these human growth hormone vials that were actually given to individuals, and that can trigger the whole picture of Alzheimer's disease. So we looked at historical exposures, things that were given prior to 1985 and now have very, very long incubation times to presentations. But I think it justifies further vigilance and surveillance of these exposed cohorts. And the other thing I remark is how interesting it was, how firmly that many in the field pushed back at these prion concepts marching into the Alzheimer's disease space. And I think one of the reasons for that is about this scary, sinister idea of CJD and prion diseases. And I think it's very important that we've emphasized in every paper that we've written and in public communications reassurance about person-to-person contagion, which is not an issue for any human prion disease either. As I've mentioned, for human prion diseases, most of those just occur sporadically, and we don't have examples aside from in the genetic cases of more than one case in a family where so there's no evidence that prion diseases can be spread by close contact, even intimate contact, and neither would there be an expectation of that here either. But I think also it's not necessarily entirely just a historical problem too, and although I'm not going to discuss this at any length at all, we note a really interesting epidemiological study based on Scandinavian blood transfusion databases. Now, this was not done to look specifically for prion risks in blood transfusion. It was done across the board to look for possible transmitted diseases in blood. But one thing that came up is brain hemorrhage, of course, which is, of course, that being a surrogate for cerebral amyloid angiopathy. So blood donors that subsequently developed multiple intracranial hemorrhage suggestive of CAA, they transmit that risk to the blood recipient, who then has an increased risk of brain hemorrhage as well, suggesting that, well, maybe actually there is a beta seeding causing clinical disease that's happening right now in the blood services. So I'll finish there. But in particular, I really want to highlight a young neurologist, Gargi Banerjee, that has really driven most of this work. And although I lead the clinic, she's been the one that has been driving the investigation and the reporting of these cases. And my director of the institute is Professor John Collins, who many of you may know. and this is where we work. Thank you very much.
Thanks to link prion, to link CAA, that is important, and maybe the last that you put on fire is to, from transfusions and blood donors, not linked to AD, is linked to the CAA. Thank you. Thank you. Please.
Yeah.
Beautiful talk. Thank you.
My timer. Hi, everyone. Good morning. Thank you very much. I'm really grateful for the opportunity to be here today. My name is Amira. I'm instructor at Stanford University and incoming assistant professor at Tufts University Medical School. And it's really a pleasure to be here today to share with you some of my most recent work about the ability of a small molecular ligand to provide resilience to synapses in a tauopathy mouse model. So I sorry. ¿El de abajo? Vale, perdón. So as we have heard earlier this morning by our colleague Alberto, the importance of synapses in neurodegenerative diseases. So synapses are very specialized structures in the brain that allows the transmission of signals, and they are also very vulnerable to pathology. That's why synaptic loss, it is a strong correlate of cognitive decline. And already in 2002, Denis Selko, one of the leaders of amyloid pathology, described Alzheimer's disease as a synaptic failure. And there is also evidence of synaptic loss as the strongest correlate of cognitive decline, even stronger than neurofibrillary tangles or amyloid pathology. And very interesting also, there is synaptic density in human studies with neuroimaging showing that synaptic density is also a very strong correlate of cognitive decline. So it's a predictor of cognitive decline in neurodegenerative diseases. So using this as the main foundation of my work, I'm going to focus today on the tauopathy synapse. So in tauopathy, toxic forms of tau pathology impairs the ability of synapses to communicate, and ultimately impairs, we can measure in the lab, in preclinical mouse models, long-term potentiation, which is a form of synaptic plasticity, like a cellular correlate of learning and memory. And this long-term potentiation underlines the transcription of newly synthesized mRNA and the synthesis of new proteins. So toxic forms of tau, as I was saying, impairs the ability of this synaptic transmission that underlies learning and memory. So sorry, I keep going down. So one of the candidate mechanisms of this tauopathy related synaptic failure could be through the modulation of P75 neurotrophin receptor signaling. And we know that because P75 receptor is relevant to human Alzheimer's disease, and there are different pieces of evidence for that. So, for example, there is colocalization of phosphorylated tau and P75 receptors in the entorhinal cortex of patients with frontotemporal dementia. And also, there are GWAS studies showing linking the P75 receptor complex to Alzheimer's disease. And also studies showing that elevated brain CSF levels of pro-NGF that augments P75 degenerative signaling is found in Alzheimer's disease patients and also tauopathy patients. And very recently, and I'm going to show some of the results in a little bit, there is a clinical trial, a phase 2 clinical trial using a modulation of P75 receptor and showing efficacy in slowing down cognitive decline in Alzheimer's disease. Therefore, since P75 degenerative signaling contributes to tauopathy synaptic failure, we have demonstrated in the lab at the molecular level some of the mechanisms by which tau pathology triggers degenerative signaling. And we've seen, like others, many other labs as well, we confirm that tau pathology increases, for example, ROE, peak of filling, and many of the kinases that leads to different post-translational modifications of tau. For example, cleavage, phosphorylation, acetylation, misfolding, oligomerizations, all of these, ultimately leading to synaptic dysfunction and the impaired ability to learn. So we see in the lab that when we apply a small molecule ligand that modulates P75 receptor, we see that modulating P75 receptor actually counteracts tauopathy-related synaptic failure by reversing all of these effects. For example, in the green arrows are everywhere where P75 receptor modulation is counteracting. And ultimately, we don't know any other studies that show that a treatment can counteract all of the post-translational modifications of tau pathology, all of them. And ultimately, and as I'm going to show in this study today, we also restore synaptic dysfunction, and I'm going to show some of the mechanisms by which synaptic dysfunction is restored by this small molecule ligand. But very briefly, I'm going to give you a brief introduction on the clinical trial that has been performed led by my PI, Frank Longo at Stanford. And this phase 2 exploratory trial using C31, I'll call it C31 from now on, in mild to moderate Alzheimer's disease, the goal was to assess safety as a primary endpoint and also a biomarker exploration of mechanism engagement. So they see, and this is the first time that we see a target that is being modulated that is other than tau or amyloid, but we can counteract both pathologies. And we see when they look at all of these different five biomarkers, they see a significant correlation with cognition. So not only a structural MRI and FDG PET, with tau being decreased and slowing down cognitive decline, but also they look at CSF biomarkers, and they see a significant correlation with cognition and decreasing inflammation and increasing synapses. And more recently, and these two last biomarkers are not published yet, but I wanted to mention to you to show the importance of synapses. So they did a proteomic studies using CSF from these patients, mild to moderate Alzheimer's disease, and they see that C31 in these patients was able to affect at least, I think it was 240 proteins that were associated to synapses. And also with correlation with cognitive decline. And very importantly, as our friend this morning explained, plasma p-tau217, that is the gold standard biomarker for tau pathology, we also see a decrease in this biomarker and associated to improve cognitive performance in these patients. So then we wanted to back translate to the mice, because all of this data motivated understanding deeper mechanistic work and understanding how synapses are affected by C31. So then in the lab, we use a mouse model of tauopathy, PS19, and we see that when we treat in vivo with this small molecule ligand, C31, we can reduce tau pathology, as you can see on the left side, in the left panel, and also we can decrease synaptic degeneration. As our colleague Alberto explained earlier, dendritic spines are key marker to understand synaptic plasticity and learning and memory. And we see that in tauopathy, compared to wild type, there is a decrease of these dendritic spines, but with the treatment, as you can see here, we can restore dendritic spines. So now the goal of my study was to really understand what are the mechanisms by which modulating P75 receptor with C31 is able to restore synaptic failure. So more specifically, mechanisms that are associated to synaptic stimulation-dependent gene and protein expression. So to do that, I use a mouse model of tauopathy P301S, and I started treating with C31 at 6 months of age. So at a time point when pathology was very well established, there was synaptic failure and neurofibrillary tangles, and then we treated for 3 months, these mice via oral gavage daily to make it more translational, since we have this wonderful data with biomarkers in patients. And 24 hours after the last dose, meaning the drug is not present at the synapses at the time of testing, I dissected the hippocampus out of the brain and collected hippocampal slices. Hippocampus is the most vulnerable region to Alzheimer's pathology and is also the center of memory storage. And then I performed electrophysiological recordings to understand synaptic function by inducing 3 events of theta burst stimulation, which is a very strong protocol, but in these mice, since they are very old and also the pathology is very well established, we need strong protocols. And then I wanted to show this because this is the 3 rigs, so multi-electrode array systems that I built independently at Stanford. And when we did this, the challenge was, can we integrate electrophysiology to understand synaptic plasticity and gene expression and protein expressions that depend on this synaptic plasticity, not separated, but really that is integrated. So to do that, I use, can the video play? Well, so to do that, I use the mouse hippocampus. This was a video showing how the mouse hippocampus is a very large structure in the brain. So we dissected the brain, the hippocampus out of the brain. No, it's okay, no problem. And then these are the hippocampal slices that ultimately results in the middle of the screen. And then we recorded synaptic plasticity from CA1, the Schaffer collateral. So I'm going to go step by step. So we confirm what other laboratories have shown before. So in the red line is tauopathy mice. And when we compare with the black line, wild-type mice, we see that synaptic plasticity is impaired. So the ability of the synapses to be strengthened is reduced compared to wild type. This has been shown before at 6 and 9 months of age in these mice. But very interesting, we find that 3 months of treatment in vivo, and the drug is not present at the synapses, so there are changes, structural and functional changes happening at the synapses at the time of testing. LTP was restored completely to wild-type level. So now it's the first time we see data with an in vivo treatment that is modulating a receptor other than tau pathology of amyloid, and we can restore synaptic function. So since this plasticity depends on the novel RNA and protein synthesis, we wanted to understand further the mechanisms. So after the recordings, I collected the slices, both after stimulation and without stimulation. So as you can see here, these are the slices at the top that did not undergo any plasticity. I used them as controls. And at the bottom, this is the multi-electrode array. So I collected both in order to assess activity-dependent profiles of gene expression using bulk RNA sequencing and also molecular biology using immunostaining. And first, we find something that I think it was really interesting, because when we compare, if you look at the, if we look at the bottom panel, here in the bottom panel is with plasticity, the top panel is without any plasticity, and then let's just look at the wild type. So wild type mice, control mice, as you can see, this is without any plasticity. So already plasticity, long-term potentiation, induced changes in the transcriptome. But then when we look at the tauopathy mice, we see a remarkable downregulation and also upregulation of genes that is found in tauopathy mice only in the context of synaptic plasticity, because here, without any plasticity, in tauopathy mice there are changes, but not as remarkable. And then with the treatment with C31 in vivo was able to restore the genes to similar patterns as the wild type. So we are seeing changes at the transcriptome level that only happens in the context of synaptic plasticity, highlighting the importance of understanding synapses during neurodegenerative diseases, and this is in the context of tauopathy. And when we look at what these genes are associated to, then we see that this downregulation in tauopathy mice were mostly enriched, as you can see here, in neurons. And these upregulated genes were enriched in microglia. So meaning there might be like an increased microglial genes that with the treatment is restored, as you can see, no more upregulation, and neurons were downregulated, and with the treatment, no longer downregulated. So let's understand the function of these genes that are so important for synaptic plasticity, and this treatment is able to restore. So sorry, this is a very busy slide, but the key message is that we see synaptic, gene ontology processes associated to inflammation here in tauopathy mice compared to wild type that so they were upregulated, and with the treatment, with C31 in vivo treatment, the same pathways, as you can see, they are here downregulated. So we are shifting the pattern of expression from upregulation to downregulation that is associated to inflammation. And the same we see for synapses. So those synaptic processes that are important for synaptic function that are downregulated here in tauopathy mice compared to wild type, we see them upregulated in tauopathy mice with C31 treatment. So this was really interesting for us to see, but then we wanted to understand, are these genes part of any modules, so gene co-expression modules that are associated to any specific functions? You know, gene co-expression analysis understands genes that co-express together because they have similar functions. So then we saw actually a large number of downregulated and upregulated gene co-expression modules in tauopathy mice compared to wild type, and with the treatment C31, all of these significance disappear. And when we look at the function of these gene co-expression modules, we see that the downregulation was again associated to neurons, so synaptic processes, and the upregulation to glial cells, so associated to inflammation and cell death. And with the treatment, all of these are restored. So C31 is able to restore synaptic function and also reduce inflammation in tauopathy mice. But what is the relevance to human Alzheimer's disease? We perform an overlap analysis to understand the database from our tauopathy mice, and we compare this database to human Alzheimer's disease existing databases to really understand whether the data we are seeing is relevant to human Alzheimer's disease. And then we see actually a very similar pattern. So we see upregulation. So this is a correlation of the genes, the tendency of the genes to be expressed in tauopathy mice towards Alzheimer's disease. And we see the same pattern, upregulation in glial, and with the treatment, downregulation of glial genes. And in neurons, the opposite pattern. So all of the downregulation that we saw in PS19 mice with a similar tendency to Alzheimer's disease, with the treatment is upregulated. So then we understand that our work is relevant to human Alzheimer's disease, and the genes that we are seeing changing is also relevant to changes that occur also in Alzheimer's disease, in human Alzheimer's disease. And finally, I'm going to just show you 2-3 slices showing at the protein level. So we wanted to confirm at the protein level these changes. So first, we look at tau pathology because we are using tauopathy mice, so we wanted to know whether the treatment also reduced pathological forms of tau. And then we see that in the slices that underwent plasticity, this is the region, I'm marking here the region where we place the electrodes and induce plasticity. And we see that ATA, the pathological form of phosphorylated tau pathology, was increased in tauopathy mice, and with the treatment, it was decreased. And also, we look at a GluA1 receptor because one of the genes that we found associated to synapses that was downregulated in tauopathy mice, and the treatment brought this gene up, it was a gene that encoded the protein glutamatergic GluA1. And then we see actually, these are also slices that underwent plasticity, so all of this is activity dependent. And then we see in the controls versus the transgenic, that you can see the decrease in the signal of GluA1 receptor. And with the treatment, it's increased again. I don't know if you can notice the difference, but basically the treatment is allowing LTP to increase the trafficking of GluA1 receptors to the postsynaptic dendrites to maintain synaptic plasticity. And finally, and this is a very recent work, very preliminary data, we just finished analyzing with my student, we see that microglia engulfment of synapses, which is a key mechanism of the degenerative signaling in tauopathies and also Alzheimer's disease, we see that with C31, let me go step by step. So in tauopathy mice, see this microglia, reactive microglia there was an increased engulfment of synapses by microglia because it's very reactive. And with the treatment with C31, this is down. So we are also helping some of the mechanisms associated to microglia. This was very relevant for our study because, as I just showed, we see microglia and neuron interaction at the transcriptome level, so we wanted to confirm at the molecular level as well. So to conclude, and with this I finish, so in this study today, I show you how in vivo treatment with C31 restore synaptic plasticity in tauopathy mice. There is also activity-dependent gene networks that are disrupted in tauopathy mice, and with C31 we can counteract these networks, and not only in the mice, but also with overlap to human Alzheimer's disease. And there are cellular mechanisms that align with plasticity rescue. So I just showed you the increased GLUA1 trafficking to the dendrites, the reduced ATA signal, and also the restored microglia synaptic engulfment. And then I've been thinking a lot about this because there is a mechanism to the clinic, but then also back translated from the clinic to the mice, to really understand how these synapse-centric effects are really in coherence with the phase 2 clinical trials that I just showed you, the data with human patients showing the effect that the synapses, the inflammation, the proteomics, and also the effect in tau pathology itself in human patients, which actually track cognition, which I think is very important to understand. There is no other treatment in clinical phase 2 starting now, is starting phase 3, and we actually have the pill ready to give to patients that targets other thing than tau or amyloid. Is targeting P75, and we are seeing a rescue and slowing down cognitive decline and restoring some of the mechanisms that are associated to the degenerative signaling. So the take-home message is P75 modulation is a translational synapse-centric strategy that actually stabilizes tauopathy circuits. So in the patients are Alzheimer's patients, but my work is really proving how these mechanisms can work in tauopathy circuits, from molecules to relevant synaptic plasticity, and really map the whole human biomarker directions that will be very essential to understand the future directions of this treatment. And with this, I want to thank you all for your attention, and thank also my lab, the Longo lab, and my PI, Frank Longo, for all his support. And he also gave me some of his slides for the introduction today. And Patricia Moran-Losada for helping with the analysis of the transcriptomic data. And yeah, thank you all for your attention, and I'll be happy to take any questions. Thank you.
Thank you so much. Thank you, Amira. Thank you, Amira, because in every one of these talks, you are involved in the new pathways with the CA, the new pathways with this study with Longo, Antonio Wiscores, which is fantastic, biomarkers, and to understand more and more the synapses, and to find a new treatments. We have only less than 10 minutes to discuss. Less than 10 minutes. I have one option. We have an option. It's something of you wants to start and ask one question, or that is open for the audience. If the audience have a question, please, very short, very concrete, and we have 10 minutes. Up to you. If in the audience have questions, here we have the answers. And if not, we can, no, I don't know if we have the answers, but we're trying to answer the question. If not, we can move with the panelists. Feel free. Option for the audience? Somebody? Because we have genetics, we have CRA, you have tauopathies, a lot, the new treatment, and the role of the biomarkers. Okay. Pasqual, tenemos 10 minutos porque hemos empezado tarde y yo soy una pésima moderadora. Una pésima moderadora, perdonar. Pero era muy interesante, ¿ok? Pasqual. With a mask.
Come in.
It's not here.
Now?
Now.
Okay, perfect. I would like to know the opinion of each of you now.
Now.
Okay. Regarding what you think would be the best strategy to deal with the tauopathy in Alzheimer's disease. Let's focus on Alzheimer's disease. So there are several options. We'd like to focus more on the clinical arena, that now that we have several clinical trials going on. I would like to know your opinion, what things you think are going to work or other things that are not going to work.
Mm-hmm. Good.
Everyone? One. Alberto?
Okay.
No, I think the Alzheimer therapeutic field is still in its infancy, so I think we will start really to see many therapies in the next years, and I think tau therapies have been challenging. I think so far the most promising is antisense oligonucleotide seems to be the most promising, but of course as a clinician this will be a huge problem in the implementation because intrathecal therapies will be very difficult to really implement in the clinic. But as a proof of concept, I think they are key because it means that there is a way to really remove tangles from the brain. And of course, that will be we need better ways to deliver these therapies in the future. And regarding tau immunotherapy, I personally think that we need to work more in the delivery better, how to direct these antibodies to neurons. And I think there was a very nice presentation in Toronto with Denali with the different brain shuttles or different ways to really combine the antibody with a vehicle to really direct the antibody towards neurons that are the target. So I think in the future, like immunotherapy seems to be a good way to go, but if you improve the delivery, and I think that will be extremely helpful for young patients, for example, that have high tau load and probably will benefit less from anti-amyloid therapies.
Another question? I have a question for you, Simon, for you and for everybody.
Yes.
Because really, it's for me so interesting that you are talking about the CAA as a product in the middle of Alzheimer's disease. And one of the questions is when we treat with MAPS, CSA, it was increasing in the world of the clinicians because it's only from the vascular diseases, no? This is increasing. One of the things is how is the link to split A-beta and how could be so interesting in order to avoid the CAA as adverse effects, because in the treatment anti-tau, treatment antibodies, the same adverse events appear in the clinical trials. And do you can link something here, because CSA is a big problem, a big effect, and it's quite difficult to modulate and to understand a little more.
For sure. And I will answer that, but maybe I could just come back to Pascal's question first, because I just want, I'll say two things from a prion point of view, okay? Vulnerabilities and strategies that we have in mind for prion diseases that seem to be working in mouse. And the nice thing about prion diseases in mouse, we know exactly it's the same disease. So if it's working in mouse, it's probably going to work in human. Two vulnerabilities, I think, for tau. You've got to express the protein. So I think gene therapy looks really promising for me. It's extremely promising in prion, and there are lots of companies coming in with platform technologies that are not expensive to adapt to different proteins. Second vulnerability, I would say, it's got to spread between neurons, and that seems to me a key place where you want to try and catch it. But coming to your question, CAA, it is indeed becoming center stage, is it not? Particularly with side effects of amyloid beta immunotherapy, and I think I would come back to Alberto, the therapeutic strand you raised, which is these shuttle technologies. And I think the ability to get amyloid beta targeting treatments into brain parenchyma with minimal vessel involvement with the kind of shuttle technology is a really appealing way to minimize some of the inflammation that seems to come about through targeting amyloid in the vessel itself. So, I think like most of us, we're especially interested to see the results of those trials, larger samples, whether the incidence of ARIA is going to be reduced using this kind of technology. And then, if that's working, adapt that technology to tau targets and others. Yeah.
Okay, thank you so much.
I have just a question for Simon. Do you have any explanation for this increase in sporadic CJD that you showed?
Yes. Thank you for the question. I think we're just getting better at diagnosing it.
Okay.
So two things. One is MRI, which is a wonderful diagnostic assay with positive features in CJD. That's cortical ribbon, high signal and diffusion weighted imaging in the basal ganglion, the thalamus, 95% sensitivity and very specific. And then the CSF RT quick test, which essentially, I'm sure you'll know, but for those in the audience, recapitulates the prion phenomenon in a test tube where you've got recombinant protein, a prion seed, you shake it and incubate for a day or so, and you get this massive conversion of the recombinant protein into the amyloid form that can be detected with dyes. And this is a remarkable diagnostic biomarker. Literally between us internationally, we can count on two hands the number of false positives out of tens of thousands of cases. So sensitivity specificity is truly 100%, I think, or with rounding error, and sensitivity about 95%. So I think diagnostically we've just got much better. I think we're still missing cases, and I think there's further to go, but I think that explains the rise, because it's happened across lots of countries, even those countries which haven't had BSE exposure.
You have another question?
Yeah.
Okay,
No.
Amira. No, you.
No, I thought that maybe I could answer to Pasqual's question and then ask a question as well. Is that okay? So very briefly to answer your question, Pasqual, in my opinion, I think that as I was showing earlier, the importance of synapses and helping synapses to stay stronger and to be resilient to pathology. I think that is one of the key strategies that the field should be more focused on in tauopathies. And as I've been showing, and there are also the work from other people showing how there is different signaling from tauopathy, so triggered by tauopathy, that underlies also the signaling of P75 receptor. And this is just one example, but maybe we should just start thinking beyond tau or amyloid in order to treat these diseases, particularly tauopathy, and really help the signaling and the synapses to function better. That's to answer your question. And then very quickly, I wanted to ask Alberto, because your work, it was very exciting with these synaptic contacts in human patients. And I wanted to know whether you have correlated those changes with different stages of pathology in these patients. Is there any difference as the disease progresses, or?
Yeah, so that is a very good question. It's difficult to address because in the brain, we work with the brain bank in Barcelona, in Hospital Clínic i d'Ibabs, and also with samples from Edinburgh. And almost 95% are very late RAG stages. Okay. So we have we have no access to really these cases with less pathology. But then in our experiment, we look at regions with less tau pathology, and then we saw that there was oligomeric tau in synapses already, like the primary visual cortex, where you have a low tau pathology.
Oh, okay.
And also we look at we didn't have time, but also in this study, we look at the synaptic loss around the different lesions. So around the tangles, you don't see much synaptic loss. So that means that synaptic loss is very distant from the tangle. But in dystrophic neurites that contain lots of tau, then you see around some damage, which is probably also because A beta is around. But we don't have really a full picture of different stages.
No, but this is very interesting. Thank you.
I have a question for you and a question for everybody. If you consider that to rescue the role of the synapses, because during all the time the role was to the deposit of beta, even that include the errors in the synapses. If we risk out the role of the synapses in terms of cognition related to the first step or the key point from the cognition, do you think that it's possible to mix, to treat patients with some drugs that it's involved directly in the synapses, and to add an anti-MAB to it's involved in tau, it's involved in beta? I don't know. That is a brainstorming, because every time we are increasing in terms of the drugs, and we are increasing to the knowledge to attract Alzheimer's disease with the different pathways. No?
I think that the future, in line with Pasqual's question, is the combination therapies, and probably in different stages of the disease, we will do different treatments. Of course, we have to start with anti-amyloid drugs. That's what will happen. We have to clear amyloid, but once we clear amyloid in the brain, what? Probably the propagation of tau have already started, so we have to stop that, and that's why we need tau antibodies. And yeah, obviously, if we have other targets, glial targets, synaptic, that would be great. Here, of course, I'm biased, but here that's why also biomarkers are so important, because that can speed up all the trials, because we have to show, we cannot wait until the end with the final outcome, which is cognition or the clinical or the functional outcome. So if we can demonstrate that each of the treatments have a target engagement, we have seen it with the tau treatments, with the removal of tau in the tau PET, and each of them we see this target engagement. This will accelerate a lot of the trials. And also regarding the tau treatments, as a target engagement, I think it's key not only to show that are able to remove tau with the tau PET or some fluid biomarkers like MTBR that are indicated in soluble tau, but the downstream effects of that, because maybe you remove tau, the tangles, but nothing else happens. So maybe synaptic biomarkers here can play a role to say, okay, we remove tangles, but is there a downstream effect of that?
Thank you. Okay.
Do we have time for one more?
I don't know. We have
Short.
time, please.
I was going to ask Marc a question. Oh, okay. No, there's one from the audience. That would be better.
You.
Yeah.
Can you hear me? Yes.
Yes.
Hi. First of all, I would like to thank all of you for your presentations. Excellent as always. And I would like to play a little bit of devil's advocate here. I'm sure that you're all aware of this population in Colombia with the PISA mutation, and the recently discovered protective isoform of ApoE3 Christchurch.
Yes.
So interestingly, and the anatomy, the pathology of this patient, showed I'm not sure whether it's published or just recently I heard it in a meeting, that this patient presented with very high amounts of A beta, increasing amount of plaques, and even more interestingly, an increase of phospho-tau 217 in blood, which actually challenges now, our conception of whether, these biomarkers, highly used biomarkers, correlate to the clinical history. So I would like to know your opinion about it, and especially, Marc and Alberto, whether you will think that perhaps, as you mentioned, could be a matter of like deposition, localization of this tau during the progression of disease. So, thank you.
No, I have to say that it's extremely interesting, the line of research of resilience. So that is a clear case, someone that has a autosomal dominant genetic form, so she or he must have Alzheimer's disease, and actually develop Alzheimer's disease, but much more delay, but has in that case a genetic protective factor that delays all the, so the resilience to the primary pathology, amyloid and tau, it's how we can modulate that is extremely interesting. But also because we can learn a lot about this. For example, a good example is the biomarkers. In that case, exactly, so there is amyloid. Phospho-tau 217, it's not a marker of Alzheimer's disease, it's a marker of the tau-related response of amyloid. And that patient had amyloid, it is resilient to the effects, the cognition is resilient to the effect of amyloid and does not have the disease, but phospho-tau 217 is indicating what it is, which is this amyloid deposition. Not only this, and this is important for the monitoring, if you want to use phospho-tau 217 as a monitoring biomarker, we don't see that does not really decrease, it does not change. So that's also a learning if we want to use phospho-tau217 as a monitoring biomarker, doing longitudinal measurements in patients that are treated. We have seen in the trials that it goes down, but it is well correlated enough for phospho-tau217 with amyloid deposition or the cognition, so we can take decisions at the individual level with the blood biomarker to stop, to restart the treatment. I'm not sure we are there.
We have the last one for you, Simon, because you want to comment something more.
I wanted to ask Marc a question about his biomarker work, and it seems to me it's starting to become very mature, the phospho-tau biomarkers, and I'm interested to know, in your locality in Barcelona, have you heard reports back from clinicians about its impact on their practice? Is it making diagnosis earlier? Are patients and families reporting benefits from receiving an earlier diagnosis? Has it got to that stage yet, or is it too early?
Yes, so we have a PhD student, Isabel Estragués, who is doing a PhD on that. So what she has done is to follow the example of AMIPAT or the IDS study with amyloid PET. What we did is to do kind of a trial with an early disclosure of the biomarker and a late disclosure half a year later, and see whether this changes. And what we see is not only that there is an increase of the diagnostic certainty by the neurologists, but we also see is that we start treatments, anticholinesterase inhibitors, earlier. We have also some questionnaires to the patients about satisfaction, and they get better information, so they understand the disease. We're analyzing the data, but it's true that it's not only about these areas in the curves. We have to study, and it's not yet well studied, whether the biomarkers have an impact in the real scenario. So we need more clinical utility studies.
Thank you.
Okay, thank you so much, everyone. The time is finished. I believe that some people punish me immediately, but thank you for attending this panel. It was so exciting panel, and for all the audience that I believe that we are learning, and it's possible to teach. Thank you very much. Okay.